Hello, I'm Sarah Jo Head from Mr. Premac's 715 Anatomy class here at Grand Canyon University, and I'll be doing my research project on mitosis and the cell cycle. Today I will be talking about the cell cycle and the details of mitosis. The first process of this is going to be interphase, and where the cells prepare for cell division. Within interphase, DNA copies itself and turns into chromatin. The chromosomes cannot be seen yet because they are loosely coiled. The DNA replicates, the cell grows, and replicates the organelles. There are 3 steps to interphase, which include the cells producing protein and other cytoplasmic organelles. G1 is going to be the first phase, which is the first gap. This is where the cells grow while the chromosomes duplicate. The next step is going to be the S phase, and this is synthesis, where the cell just grows some more. After that, it goes into G2, which will be the second gap, and it is ready to divide for mitosis to begin. The cell division of mitosis is a process of many steps until the 2 daughter cells are made. Now that interphase is done, we'll move on to mitosis. The first step of mitosis is going to be early prophase. Early prophase is only found within plant cells. This is where the chromosomes coil together tightly and can now be seen. This is also where an array of spindle fibers or microtubules extend between each centriole pair. Spindle fibers or microtubules and centrioles. Astral pairs are just like spindle fibers, but smaller, and they radiate into the cytoplasm. Late prophase is where the nuclear envelope disappears, the chromosomes coil up and spindle fibers are formed, and each copy of the chromatid will then connect and match to its pair. Now that we have moved into late prophase, the nuclear envelope have disappeared, chromosomes are coiled up, spindle fibers are formed, and each copy of the chromatid will then connect and match up to its paired chromatid. The location that it connects is going to be called the centromere. Kinetochores attach to spindle fibers and are the protein-bound area within the centromere. Metaphase is the next step. Within metaphase, the chromosomes that are now paired up with one another are going to line up in the middle. Just like that. Now that they are paired up in the middle of the cell, or the metaphase plate, the spindle fibers are going to connect to the chromosomes. Spindle fibers have connected to the chromosomes. We'll move into the next step of mitosis, which is anaphase. Anaphase means to come apart. So during this step, the chromosomes copies will divide and the spindle fibers will pull chromosomes to the opposite poles. These are now called the 2 daughter chromosomes. Now that we have moved into the 2 daughter cells, it is time for the final step of mitosis, which is telophase. Telophase is where the spindle fibers disappear, the chromosomes split apart and uncoil, nuclear envelopes form and 2 new nuclei are formed. As soon as telophase starts, so does cytokinesis. It is when the cells start separating and the cytoplasm pinches in to create a cleavage furrow. Cytokinesis is the division of the rest of the cell, cytoplasm, and organelles, and the nucleus divides. Within animal cells, the cytoplasm would pinch in, and in the plant cells, the cell plates would form. Once cytokinesis is done, so is cell division.